Welcome everyone to your own channel, ADP for Learning. I am your own learning partner cum host, Ashish. So, let's begin our learning journey. Today, we are continuing our learning journey about the Interactive Glossary of Genomic and Genetic terms. And our motto is, 5 terms at a time, keep the learning in the mind. Starting with the first term, that is cancer. Cancer is a disease in which some of the body's cells grow uncontrollably. There are many different types of cancer, and each begins when a single cell acquires a genomic change or mutation that allows the cell to divide and multiply unchecked. Additional mutations can cause the cancer to spread to other sites. Such mutations can be caused by errors during DNA replication or result from DNA damage due to environmental exposures such as tobacco smoke or the sun's ultraviolet rays. In certain cases, mutations in cancer genes are inherited, which increases a person's risk of developing cancer. Importantly, most mutations that occur in cancer genes don't actually lead to disease. Your body has an excellent system of DNA repair genes that actually remove most such mutations as they occur before they can be propagated. The second related term is cancer susceptibility gene. A cancer susceptibility gene is a gene that, when changed or mutated, gives an individual an increased risk for developing cancer. Individuals who have inherited mutations in certain cancer susceptibility genes have a lifetime risk of cancer that is significantly higher than the general population. For example, BRCA1 or BRCA2 genes. Individuals in the high-risk category may benefit from more frequent cancer screens. There are also many gene variants associated with a small increase in risk. In some cases, environmental factors may also play a role. One of the most interesting research questions in cancer susceptibility is why certain genes predispose to particular cancers. For instance, women with mutations in the BRCA1 or BRCA2 genes are at increased risk for cancers of the breast and ovary, but not for blood cancers like lymphoma or leukemia. The third important term is candidate gene. The term candidate gene refers to a gene that is believed to be related to a particular trait, such as a disease or a physical attribute. Because of its genomic location or its known function, the gene is suspected to play a role in that trait, thus making it a candidate for additional study. The more you know about a trait, the better job you can do selecting a candidate gene for further study. For instance, if you're studying the genetics of body size, genes that control bone growth, lipid processing, and insulin growth factors are all excellent candidate genes. The fourth term is also related to first 2 terms mentioned before, that is, carcinogen. A carcinogen is a substance, organism, or agent capable of causing cancer. Carcinogens may occur naturally in the environment, such as ultraviolet rays and sunlight, and certain viruses, or may be generated by humans, such as automobile exhaust fumes and cigarette smoke. Most carcinogens work by interacting with a cell's DNA to produce mutations. A carcinogen is any substance that can cause cancer. It's important to identify items that might be carcinogenic because we can then take specific measures to avoid or limit our exposure to them. The U.S. Department of Health and Human Services National Toxicology Program and the World Health Organization's International Agency for Research on Cancer both use evidence-based approaches to catalog substances that are known or reasonably anticipated to be human carcinogens. Both use evidence-based approaches to catalog substances that are known or are reasonably anticipated to be human carcinogens. To date, over 500 substances have been identified as definitive, probable, or possible carcinogens for humans. This includes items like asbestos, automobile exhaust, processed meat, or ultraviolet rays. I should stress that exposure to a carcinogen does not necessarily mean you will get cancer. A number of factors influence whether a person exposed to a carcinogen will ultimately develop cancer, including the amount and duration of the exposure, exposure to other environmental factors, and the individual's genetic background. The fifth most commonly used term is carrier. A carrier, as related to genetics, is an individual who carries and can pass on to its offspring a genomic variant or allele associated with a disease or trait that is inherited in an autosomal recessive or sex-linked manner, and who does not show symptoms of that disease or features of that trait. The carrier has inherited the variant allele from one parent and a normal allele from the other parent. Any offspring of carriers is at risk of inheriting a variant allele from their parents, which would result in that child having the disease or trait. The key to understanding carrier in the genetic sense is that, even though we all have 2 copies of each gene, For some genes, you can have only one working copy and essentially have no major medical issues. The challenge arises when you have a child with another person who is also a carrier for the same autosomal recessive disorder, and the child inherits both non-working copies from each parent. There's a 25% chance of this happening with every pregnancy. Most often, people don't know that they are carriers. As a result, it's now possible to genetically screen prospective parents to determine whether they are carriers for any of the more commonly seen autosomal recessive disorders. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.